So we were interested in understanding how much those how much that metabolism change from the dry period all the way to early lactation, and then track those changes that see if there are any changes on metabolic function in the transition period.
Welcome to the Fit Science podcast brought to you by Wisenetics. I am Carlos Campabadal from Kansas State University, dedicated to bringing you the latest insights and discussions in the fit science from the fit science industry. Please get ready to expand your knowledge and stay ahead with today's episode. So today it is a great pleasure to introduce our guest today, Dr. Lautaro Rostol. Thank you very much for joining us today for our podcast here from the Fit Science podcast from Wisenetics. In order to start, first of all, welcome you, and you please just tell us a little bit about yourself and how do you end up where you are today?
Yeah, thanks. Thanks for inviting me. This is always a pleasure to be here. I'm originally from Argentina, as we were discussing before starting recording. And I studied there, I did my undergrad there, and then I moved to the US, did a master's in Florida, at the University of Florida, with Nicholas Di Lorenzo, in beef cattle, actually, on wean calves, like during their transition and like looking at immune function in response to a challenge like they were using as a model back then. And then did my PhD in Canada at the University of Guelph, also working in calves, like this time in dairy calves with Mike Steele at the University of Guelph. Spent there almost 4 years. And then at the end of my PhD, I had the opportunity to apply to this position here at University of Madison, and I've been here since the beginning of 2023. So it's been almost 2 years since I started this position. So the position is on bovine immunology. So I work now both in dairy calves and in cows in the transition period. So this area on the transition has been fascinating. We did some work on that during my PhD, but I've been largely expanding on that area since I started here with collaboration with various people here in the department, mainly Dr. Head.
Great. No. Thank you very much for your introduction. And we know that you have a great career going from, I see it going south all the way north, in transitions,
We have we had a joke with my wife saying like this is the furthest north we're gonna move, like when we're in Canada and Guelph, and it's like well, let's try to. This is the furthest we're gonna go. Yeah.
So yeah, we're going from, Argentina, Florida, kind of like in the middle, and then Canada, and now you're making your way down to Wisconsin.
I think the biggest transition was Florida to Canada.
Yeah, I bet it was. Yeah.
So warm to so cold. But yeah, from Guelph to here is barely almost the same. Yeah.
I could see that. And Wisconsin, I mean, for many places, and then you do research in the field with cows, it is cold compared to a lot of the places here in the United States, but great transition. So for our audience, please subscribe to our channel and click on the bell to receive any notifications. So Dr. Rostol, very interesting of your work, especially in beef cattle and this and also on dairy. So I know you have done a lot of your work in transition cow health and just overall changes in immunity. So can you expand a little bit what you're working right now and tell the audience so they can learn a little bit about what you do?
Yeah, absolutely. So one of the areas that we're working in here is trying to really understand from a mechanistic perspective, what happens to the immune system during this transition period? We've known for a long time that cows suffer from a lot of different health-related disorders during this period of time that spans from 3 weeks before calving to the first 3 weeks after this cow's calf. There's a lot of things that happens that needs to happen for that animal when it calves. There's physiological adaptations. There's hormonal changes as well from those animals, endocrine changes, and one of the things that everyone discuss about is that negative energy balance that these animals have when they go from not producing any milk to producing copious amounts of milk in just a couple of days. And then those animals have to quickly adapt to that new physiological state. No, so that imposes challenges to the immune system. There seems to be increase on the inflammatory tone of the animal. The negative energy balance stage of that animal is compensated by mobilization of adipose tissue. There's changes in liver function that occurs during that period of time. So all of that together, that is a very complex phenotype to try to understand. And there's a lot of interactions, a lot of different systems, seems to increase the susceptibility of these animals to disease during this period. And for a long time, this period has been, like, people have been describing this as the immune system being either down regulated or not functioning to the level they can do either on the dry period or during later on in the lactation. So they talk about suppression of the immune system on these animals. This largely comes from studies where they isolate mainly neutrophils or monocytes, and then they in vitro they try to stimulate them to respond as they would respond to a normal pathogen. And they see that they don't respond as well in the early period post-calving compared to the dry period. The dry period being when the animal is not producing milk, right? And there's no more to it than that. There's other studies from, especially Andres Contreras has really done like really good work in the last couple of years, characterizing what are maybe the main sources of where that inflammation is coming from, and then trying to do more work on understanding how these different immune cell subsets are behaving, and then he focused on macrophages. That is another subset, and then his research like really shows that maybe there's changes on the metabolic environment for those cells that predispose them to become more inflammatory. And I think that has been really informative in like how we do our decision making on the management of these animals. And also he's done some work on trying to understand before calving what are the animals that might be at biggest risk or at more risk coming into lactation, so we can identify them and maybe try to manage them differently. I don't think we're quite there yet, but there's been a lot of work on that area. So our work so far, it's being two things: trying to understand what are some of the changes that happens in the metabolism of these cells, particularly a group of cells called T cells. And we like focusing on them because there's a group that are called T helper cells that they function. I like to say in my class, in my animal health class, that they are like the coach of the immune system, if you want to use a soccer reference. And they integrate a lot of the signals from the environment, and then they dictate what type of immune response they're going to command. So like they really function as a hub of understanding what are like the different cues of the environment, and then they respond in various ways to depending on the specific threat. So that system has to be really well fine-tuned in order to respond properly, but it's obviously influenced by external signals like hormonal changes and metabolic changes. So we were really interested and said, okay, can we map or track if there are changes on their metabolic function during this transition period? So we use an equipment called a seahorse that lets you see oxygen consumption rate of these cells, and then another pathway for energy production that is called glycolysis. And then something very characteristic of immune cells is that when they recognize a pathogen or an antigen from a pathogen and then they become activated, they use a lot of energy to respond properly. mainly because they rapidly start expanding. So like once the cells become activated, they clonally expand. So in that process, they use a lot of energy and they need it really quickly. So instead of using oxidative phosphorylation, that is the main pathway for most of the cells under normal oxygen levels, um, when there's no hypoxia in the environment, they use oxidative phosphorylation. But when they become activated, because they need to tap into these energy resources much more quickly, they switch to glycolysis. So even though glycolysis is less efficient in producing energy from a molecule of glucose, you access those ATP molecules much faster. And then they use the intermediaries of partial oxidation of or partial use of glucose for uh, building units for molecules they're going to use in their immune response. So reactive oxygen species, glutathione peroxidase, stuff like that. So you can track those responses, metabolic responses in vitro now at a really good detail and on a cell level basis. Um, so we were interested in understanding how much those how much that metabolism change from the dry period all the way to early lactation, and then track those changes that see if there are any changes on metabolic function in the transition period. Because metabolism of these specific cells, in a large sense, they also dictate function. There's a big correlation there. And from the human side, we know that people that has different metabolic disorders, they have this function on these specific cell types, uh, and these pathways are largely involved. So we were curious of understanding. So the first study was just a short collaboration with Heather White, just tapping into some of these questions just to develop the methodology. And this was on mid lactation. And then once we had that figured out and everything was working, we did this study where we follow them all the way from the dry period through early lactation. And then the other thing that is important, like to give a little background, is mitochondrial function is really important, specifically on normal oxidative phosphorylation. And then when there are increases in metabolic demands, like sometimes that those mitochondria can be pushed too far. So like the mitochondria is like the engine of the cell, right? So if you push it too hard, the engine is like maybe stressed out, and that in itself can create what's called oxidative stress, and that can create cell damage, and that can also influence how these cells are behaving. Right? So that's kind of like the background. So what we observed in the study is we isolated the cells. We isolated specifically the T helper cells, and we had one group that we just observed how these metabolic changes were at a basal state. We just observed the cell kinetics over time. And then the other group, we activated them. What means is you trick the cell into thinking there's a pathogen, and then you observe how they conduct with that response. And then we compare between the different time points. So dry period, that was a month before calving, 3 days after calving, and then a month after calving. So what we observe is at a basal state, which was really surprising because we didn't think we were going to observe anything there. Actually, the cells were consuming more oxygen, so they were using more oxidative phosphorylation, and they were also doing more glycolysis. So the whole metabolic set point was higher after calving. And then once you activate those cells, they switch, as I mentioned before, from oxidative phosphorylation into glycolysis, and they're also using much more glycolysis, and that was much more pronounced post-calving compared to the dry period. And I don't remember from the top of my head, but I think day 3 and day 28 were pretty close to each other. So there's a switch. There seems to be a switch in the metabolic set point of these cells, and these cells seems to be more active, not less active. And then the other thing we did is um, we track how much the cells proliferate once they become activated. This was a different protocol in the lab um, that allows us to look at how much the cells expand and proliferate based on a, on an activation. Uh, and the same thing, they are much more active. They proliferate more uh, in the post-calving period compared to pre-calving.
And just to very important, because this is great information, and what you're doing there, for our audience, if they have any questions, please write them in the comment section, and we'll be sure to get them to Dr. Rostol, so he could answer them directly, or you could reach out to him directly. So very important in all this. But as you continue telling us the other a part of your work on that sense, can you also tell us a little bit of how that nutrition, was being something very important for our overall immune system and metabolic and on any animal at the end of the day, how does that support that health and the whole metabolic changes?
Yeah, I think we're still very early on stages of understanding this, and like we're still like trying to. That's probably going to be the next study of how we can try to modulate these mechanisms, right? And then see um, if they really are related to a specific phenotype on the animal. So, like being that, do all animals experience these same changes, or is it different for animals that have a specific um, health disorder event on the transition period versus animals that have a normal and healthy and uneventful transition period, right? So that's a next study that we're trying to put together. Also, we have just some preliminary data that through specific amino acid supplementation, you might be able to alter this, but we're still very early on to say anything conclusive. But at least based on the preliminary work that we're doing, there are maybe some nutritional interventions that could be used
All right.
to type into this. Um, I think mainly amino acid
Amino acid,
supplementation.
as more as a feed additive, synthetic amino acid, things like that.
Yeah. So we've seen even in calves on weaning, we have a poster that we'll present this year at ADSA that supplementation of protective methionine on the starter diet on calves, we can improve, because this weaning period is the closest thing you have to the transition period. That there's a lot of series of events that changes very quickly,
Yeah.
that imposes a challenge and stress on the animal that impacts on health outcomes as well. And at least on that model, we see that methionine supplementation seems to improve um, cell function in these same assays. Um, so yeah, and then another study that we conducted. This is actually in Canada, where I did my PhD. Um, in a study that we did in collaboration with them, we didn't measure the same thing on the cows, but we look at specific markers on these immune cell subsets uh, related to state of activation of the cell. So we were measuring things that are like specific proteins on these cells that are related to how much or how less they are activated on these almost the same time points. And we saw exactly the same on those same studies. So I think the main conclusion from all of this is that actually the immune system of these animals, particularly these T cells that are really important in commanding the overall systemic response, they're much more active. They're not suppressed. So what we're thinking that is maybe the type of response that they're eliciting is incorrect, and then the response is either overreacting or like tilted towards a very inflammatory response rather than the specific tailored response that is needed is like skewed towards something that is incorrect.
And well, first of all, for our audience, if you learned something new today, please share the video. This is very important information that we're getting on very high end research. But going to what you're telling us today, I'm just curious, how does the weather and of course, there's always, I always think about, and I put my engineer hat on it, how weather and external factors are affecting you in terms of animal population, the pen size, all those type of things that you could kind of tell us, to put it a little bit in context for our audience.
Yeah, absolutely. So we've been trying to think how can we like try to like start applying like some of these first concepts that are coming out of this research. And I go back for now. I just go back to the basics, and I think it's probably a pretty precarious moment for the immune system. So any stress that happens on top of that might be enough to tip the balance into one or the other. So probably things like heat stress, um, environmental or be hit like social stress
So
around this time are probably going to start adding up. But this is more an hypothesis. I want to like say that like more than an actual statement because we're still working around that, right?
yeah, I'm pretty
Uh,
sure.
but if you go back into the literature, um, one of the two things that seems to dictate the responses of these cows in the transition period the most is dry matter intake on the dry period
Okay.
and body condition score of
Okay.
these animals. So even before we start talking about nutritional interventions, those are probably the two things we have to take into account. So calving fat cows, especially on the higher end, like more than 3.5, you are more likely to have problems on those cows during the transition. So those cows are calved higher than 3.25 to 3.5. Um, they might not express it specifically in terms of milk production, but those are the cows are probably going to be at higher risk of metabolic disorders and of actual health disorders, retained placenta, displaced abomasum, uh, metritis, mastitis. That's pretty well established. Then, in terms of dry matter intake, there was a lot. There was some work, maybe 15 or like around 20, 15 years ago, of University of British Columbia,
Oh,
that they did this really nice study that they retrospectively saw the intake of cows that had metritis versus cows that had no metritis events, and then they look at their dry matter intake of those cows during their dry period, not just the drop on intake that they suffer after like right close to calving,
okay.
but like their average dry matter intake throughout their entire 60 days of dry period. What they saw, what's really interesting, is that the cows that were eating below the average threshold, those are the cows that were having more problems. So those are cows that are mobilizing more fat. They are not eating to their requirements, and those are the cows that have problem when they calve.
Okay.
Uh, versus the cows that were eating well throughout the entire period, those cows are typically fine. And what they saw is that there was a behavioral component to that the cows that were not eating sufficient is the cows that were reaching the feed bank at a later stage.
Okay.
They
Oh,
were too densely populated in the pens. So you had the dominant cows that would get in front of the feed bank, eat first, and then these other cows, regardless if they would come and eat afterwards, they were not able to reach the same level of dry matter intake compared to the other counterparts, right? So uh, actually, my colleague here, Nigel Cook, he had a podcast, like an episode on his um, um, on something that he did for an extension event, and then he was making these comments as well that I think is really important. Although like this is older research, I think it's important to drive these messages home that stocking density, even on the dry pen, is really important because you need to let all the cows eat at the same time when you deliver feed, and is that seems to be directly correlated to that the phenotype of disease incidence on the
oh,
transition.
interesting. Yeah.
Um, And then the other thing that he also said that I completely agree is stress close to calving also seems to be um, a detrimental factor for disease incidence. So try not to regroup animals close to calving as much as possible, because again, that also impacts dry matter intake. Like going back to that basic
Yeah.
concept.
Hmm.
Once you regroup animals, they are very hierarchical animals, so they have to reestablish their order again. Uh, who's the boss? Who's in the
Yeah.
middle? Right. So that seems to take about like 2 or 3 days to occur. So if you have very small groups and they're constantly regrouping animals, that on the long term is probably going to come and bite you. So try to have maybe do less regroupings, especially in the last week before calving.
Yes, I do remember that hierarchy as it gets established. I mean, I grew up on a beef cattle farm, so and dairy, so
Yeah,
it's
definitely. Even if we don't realize and we don't understand it, that's definitely there.
yeah, it's definitely there. So. And then, of course, our audience, if please give us a thumbs up, like us if you like this podcast here with Dr. Rostol. And we're coming kind of like a little bit to the end of our time, but it is important to kind of get the idea for our audience. I'm assuming you guys are going to start looking also genetics and other type of things as this research continues to grow.
Yes, and that's a great comment. We don't have a specific project, but we been in discussions with a group from Iowa State that has a group of cows from I heard that it was actually from the University of Minnesota, that they were able to keep cows with genetics from the 60s,
Oh wow!
and they compare those cows against contemporary cows, right? And there's a lot of applications you can use that for. And then, of course, first thing you see, these are smaller cows, and then of course they produce maybe 20% of the milk contemporary cows produce.
Wow, I didn't know that. I knew there was less, but I didn't know that less. So.
So that's amazing. Like the advancements of the dairy industry in the last 50 years are phenomenal. But there are probably a few things that maybe have been ignored, like during the selection pressure for milk production, that were lost in that process. And they did a very interesting study that they genotyped and compare the two groups. They did it's called a GWAS analysis. And then one of the things that they saw as this most striking difference, of course, besides production, are genes related to immunity.
Ah, okay.
And there's one specific cluster that is called MHC, the major histocompatibility complex, that is it's a molecule that is used to present antigens to cells of the adaptive immune system. That is important, and seems to the contemporary cows they have less haplotypes of that. There's a lot of um, limitations from that study because they have a small group of animals from these 60s genetics cows, but at least it's informative. So like maybe there's something that we can start seeing, that maybe there are a few things that have been lost like during that pressure for milk production
Hmm.
from the immune component, that maybe someone, I'm not a geneticist, so this is I would be happy to collaborate, but this is not my specific area. But like this is something that we've been discussing with a group from Iowa, like trying to do some studies related to that. And then they also did some interesting study that they would challenge these 60 genetics cows with a mastitis pathogen and
Okay.
compare it to the same challenge to contemporary cows, and the cows respond differently. The cows from the 60s, they're able to contain that infection just to the mammary gland, and they resolve it quicker.
Ah,
Contemporary cows, that infection becomes systemic. Not the infection, but the immune response becomes systemic, and it takes them longer to, which makes sense.
it does make sense.
Much larger surface area, is
Yeah.
much more metabolically active.
Yeah.
Uh, um, but I think, at least to me, it's interesting.
And I didn't know that they have those cows there to kind of do that, which is a good comparison to see how.
Yeah, it's really cool, and it's been really challenging for them to be able to preserve those genetics
Yes,
for
I
such
bet
a
it
long
was,
time. Yeah,
and especially now these days, and for many reasons that could definitely be another podcast. But yeah, so
yeah, and I've heard they're really feisty. So poor grad students have
exactly,
had to
I was
manage
gonna
them
say.
there.
It's time for our famous three. Evonik Animal Nutrition is committed to ensure food security and safety while reducing the ecological footprint of animal farming. Its products and services use evidence-based solutions that seek to promote animal welfare and reduce reliance on natural resources. All this is underpinned by long-standing industry partnerships and deep customer understanding. Evonik's focus on efficiency, sustainable, healthy nutrition, and collaborations with livestock farming partners creates value for customers and consumers.
Great. No. This is great information, Dr. Lautaro Rostol. So just to wrap up, we always like to tell if somebody wants to continue learning or kind of get to know more that topic that you were mentioning today, or when we're talking about just overall transition cow health. What resources or what book or what website will you recommend somebody that is not an expert that could want to learn more about it?
I think it depends. Like maybe if it's a grad student that like wants to get into immunology work, first, like it's important to understand the basics. There's some good. I use a book for class for my animal health class. It's called Veterinary Immunology by Ian Tissert. That is a vet immunology book. That's like my standard book that we check if we use for class, and then we check every so often if we want to revisit something specific of like the traditional immunology.
Okay.
And then Journal of Dairy Science
Of course. Yeah.
for transition health related research. Um, and yeah, I think those two.
And besides those, do you have any? If you're, let's say, we're talking about a young professional trying to grow their career in agriculture, do you have any book or resources or any advice, to be honest, that we recommend somebody outside of technical, books or information, something that you would tell like grad students or undergrads or somebody just starting their career?
Yeah, like there's a book I really like that I try to get some ideas and concepts from for being a mentor as a faculty mentor. Is this guy Simon Sinek, that he comes from marketing, but he has some really cool concepts of how to manage groups and how to get people to be invested in what they do, like be really dedicated and committed to their work, but like and try to generate a good environment for everyone to try to strive in. So he has a series of books, but like the main one, it's started with Why, from this guy that I really like.
Great. No, this is great information, and I think especially for our young audience, please take notes on that, and that is very important. So with that, thank you very much, Dr. Rosol, for your time and all your great explanations. And this concludes another episode of the Feed Science Podcast presented by Wisenetics. Thank you for joining us today. Don't forget to subscribe to our podcast to stay updated on any other future episode. And if you enjoyed today's episode, please leave us a review. Your feedback is very valuable to us. Until next time, this is Carlos Campaudal from the Feed Science Podcast signing off. Thank you very much.
Thank you, Carlos.